So it's a tremendous pleasure to be with you. What Ambika didn't say, and thank you very much for the very generous introduction, is that I have a long relationship with India. I've been in India for nearly 20 years, almost every year. I did 1 year sabbatical at the NCBS in Bangalore in 2011. This has been a turning point in my career. There have been many turning points. But when I see all of your young faces, it's extremely energizing. It reminds me that I was one of you some 35 years ago. And at the time, I loved biology. I love the living. I still love the living in these days, today. Back then, I was very disappointed nonetheless by the teaching of biology. I was much more interested in mathematics and physics and chemistry. And so it took me a long while in my journey into science to actually discover that I could be a scientist, that I could both do the living, study the living, and do it in a context which, as we will discuss today, was grounded on the foundations of physics and chemistry, using mathematics as a language to study the living. And so today, really, what I will try to do, my best, is to try to convey to you, one, my passion for biology. Why is it so amazing? Second, how could you, each of you, if you wish, be one of the many people who try to crack the mystery of the living, which is an endless adventure. So studying the living is an adventure. It's absolutely amazing. As Ambika said, this is a long journey like any adventure, which is made of mostly short, small failures, but big successes if you're persistent and patient. How can you be persistent and patient? If you're passionated. If you have the passion for something, then you could accept, these long trails. You could use the metaphor of walking, going in a hike. You don't necessarily see the summit because it is in the cloud. Just go somewhere. Science is not about seeing a summit and going there. It's about choosing a path that you think is exciting, accepting that you will be lost most of the times, but every step is interesting, even if it fails. Okay, so I will not have the time to explain to you all my many failures. I will try to basically convey to you more importantly the passion for studying the living. Okay, so I want to say something else about this hour and a half that we'll have together. Let's have an hour and a half, the two of us together, rather than me speaking for 45 minutes uninterrupted and then you ask questions. I want to ask questions. I want to be asked questions as I go on. And I will manage the time. I will make sure that we can go through enough of the material that I wanted to go into. And so wherever you are in the room, in the back or in the front, raise your hands and ask the questions loud and clear for everybody to listen. Okay? So let's get started. So the title of my talk is really about, the origin of forms. I mean, the living, everything I do, whether in teaching or whether in research, is about answering this endless question or difficult questions: what is life? What is the living? Okay? So one of the attributes of the living is that living matter is organized. This was recognized a long time ago, and the principles of this organization is really what is my main focus of research. And you could talk about shapes and forms. It's the idea that shapes have connected to a function, which could be referred to organization. So we really want to understand that. So let's begin with a little statement, if it is necessary. Life is beautiful. These are pictures of many organisms. You could go any place and you should be bewildered by just how diverse and beautiful life is. It's also amazing. And so I will have, in the first part of my talk, sort of going into a number of things that I find amazing or puzzling. And maybe some of you could share what you think is also puzzling in life. But let's begin with a simple question so that we could get together on some way of working. So this is a question about ecosystems and life on Earth. You know that life started probably like 3.5 plus billion years ago, okay? So life probably emerged within a few 100 million years, and it has invaded every ecosystem on Earth. So what do you think is, so you're going to raise your hands for A, B, C, or D. Wait a second. So you've read the question, what is the most abundant in mass? So who raised their hands for bacteria? Raise your hands. Okay. B, ants. A bit less. C, cattle. One courageous person, thank you. D, trees. Okay, so you vote for bacteria by and large. Wrong. We'll see the answer later on. But that was good. I know everybody's wrong in this answering this question, don't worry. Now, rank by abundance, human, fish, wild birds, and poultry. Who raised their hands for A? Human, in mass, not in numbers, obviously. Fish, B. Wild birds, C. And poultry. So fish and poultry were, I think, the principles. Well, let's see. So this is the first answer. Trees and plants are by far the most abundant biomass on Earth. Okay? Look at the animals. It's tiny down there. Okay? This is in units of gigatons of carbon. Bacteria, compared to plants, is also a small portion. So when you go outside and see the trees, just see this is the most abundant biomass on Earth. Bacteria just goes only second. But now let's go into the animals. The animals are shown here. Arthropods, of which insects are part, and ants only a tiny fraction, is the most abundant biomass of animals, amongst animals. But look at, all the wild mammals. This is not much. Humans and livestock is about the same. So you see a number of surprising things already, but you should remember that, why is it important to take care of the biodiversity is because it's not what you think it is. And when you look at these majestuous animals and mammals in India, or the birds and all of that, these are just a tiny fraction of biomass. Okay, so the ecosystems are very complex. I only encourage you to basically get into the science of facts and numbers to get a better assessment of what is at stake. Here, what is changing, you have expectations, you raise your hands, but these are the numbers. These are estimates, okay? These are not exactly accurate, but they're pretty good estimates. And so this is a way to get into science, to get the numbers correct, which makes change your mind and go on. And so this is very much the approach in science, is to be anchored on data, and by that I mean quantitative data. So, first part, life is amazing and puzzling. So I would like to enumerate a number of amazing and puzzling facts, and I want to have your reactions if you either disagree that it's amazing. So first of all, size. So the smallest animal that or not animals, living organism that we know are bacteria. Okay? So bacteria, do you know what the typical size of bacteria? Well, it's all on this slide. So it's about a micron in length, okay? A micron, if you count the volume, this is 10 to the minus 15 liter, okay? It's small. It's a picogram. It's swimming. Down there in this video, you can see Bacillus subtilis that is swimming on a thin above a substrate. Okay? The largest swimming animal that we know of is the blue whale, which you could see a picture of. And at the bottom here, you see a few sperm whales that are swimming. So if you calculate the range in size, in mass, between this bacterium and a blue whale, it's 10 to the 20th. How big is 10 to the 20th? It just gives you a number to ponder, okay? The fact that living animals have explored this ability of swimming in so vastly different sizes is really interesting. And we'll come back to what is so different as a world for a bacterium as a blue whale. It is very different. We'll come to that in a second. But if I give you this number, the scale difference, 10 to the 20th, I'm going to ask you the following question. If I'm a blue whale, and I want to consider something that is 10 to the 20th larger than me, what would that be? Raise your hands and shout. 10 to the 20th larger than a blue whale. Well, don't tell me 10 to the 20th blue whales. So give me this, the solar system. Who wants to say more or less than the solar system? Raise your hand if it's more. Less. A lot of undecided people. Okay? No. Okay. So, 10 to the 20th is about twice the mass of planet Earth. But still, come on. That's a big difference, right? That means that life have explored the swimming function across organisms that can range in size from a blue whale to 2 times the mass of planet Earth in proportion. Okay? As if you could make a swimming planet Earth, you know, or 2. So another example is if I am the moon, 10 to the 20th is about 2 times our galaxy. Okay? So this is just meant to show you that the vast proportion of size in the living, this is just amazing. Okay? Now I'm going to argue in the next couple of slides that swimming, if you're a blue whale or if you're a bacterium, is a very, very different business. Okay? So let's get into that. This is meant to be shocking because the numbers are shocking. Just as we started with the quiz, you know, we are usually wrong in our intuitions about numbers. Here you're supposed to be puzzled because it's surprising that it should be so different. Okay? So, before we get into the business of swimming if you're an animal, I want you to think through the range in size in a different way. Let's consider the smallest organism that we know of. We talk about bacterium, but we could also consider, you know, multicellular organisms and single-cell organisms. So we compare here the size of the largest single-cell organism. These are the ciliates that live in the oceans, okay? And so these are 2 examples of those, in particular, or in the pond water. The paramecium, which could be, you know, 1/4 of a millimeter in size. Some other cells could be as large as 1 millimeter or 2 millimeters. So you could see them with your naked eye, a single cell. But there are also multicellular organisms like this parasitic wasp, which is about the same size. This is all in scale. So think of a wasp which has a brain, a memory, you know, they can do sexual reproduction, they can fly, and they have 10,000 cells, okay, and they're as big as a single-cell organism, okay? So you could wonder, which is the most complex organism, the single cell or the multicellular organism, okay? What? So the multicellular organism is more complex. Yeah, maybe. It actually is, the brain is so small, so packed, that the organism got rid of its nuclei to make it even more packing. They just have the bundles of axons. It lives just a few hours to just find the mate and then it dies. So it's just, you know, meant to live very short time. It certainly has a brain and can do computation in ways that, you know, is pretty complex. Nonetheless, I want to tell you that these single-cell organisms, there are 2 examples here, are really amazing. They can regenerate, they can prey on animals or other cells, they can move, they compute also, they do very complex tasks, they have memory, okay? So I think we may have to consider the complexity of single-cell organisms in new ways. Okay, so I told you that this parasitic wasp is tiny. It has basically the same body structure or shape as a hornet, a giant hornet. If you were to put them in proportion, are you ready? I'm going to rescale them. Same body plan, same basic structure. So it's like, you know, a jumbo jet with a tiny, tiny aeroplane. Okay? But they have the same basic structure. They are, you know, much more similar to one another than most animals because they have this ancestry, this commonality in insects. So this is just a means for you to be surprised by the diversity of size, even in very similar structured animals. Okay, so much for size. Now let's go to another fundamental attribute of the living, which is movement. So cells or organisms can move as single entities or in groups, like flocks. Like when you enter the rooms, it was like a flock of you entering the room. You each found your seat. You know, you're very familiar with traffic jams, of course. So when you put too many people, too many humans, whether walking or on bicycles or cars in some place, it jams. Now, if you take many cells together at high density, do you think they're going to jam? Raise your hands. Well, either you say they jam, and then it's just like the other organisms, but if they jammed, how could you build an organism like us, which is made of millions and billions of cells which have to move past one another? in a way to structure an embryo, an organ. We'll come back to that later on. So you might get the intuition that, of course, cells have to be able to move, even slowly. So is it going to be jammed or not jammed? Well, the answer is it can be, but in most cases, it isn't the case. And so let's look at a flock of cells. And there are two examples. On the top right movie, I hope you can see, you could see a lawn of bacteria that are moving as a very dense swarm. The movie is slowed down 2.5. So that means that these bacteria, even though in a super crowded environment, they move one another at super high speed, as if it was like a very fluid environment. At the bottom, you see now a group of cells which are from the kidney of a mammal. Here it's a dog, but it could be you. And look at how they make this sort of viscous flow of cells past one another. So cells have these properties, whether they are bacteria or cells of your body, that when packed together, instead of being jammed, they can be in a fluid state. And so this is puzzling. How is it that they can be so dense and yet very fluid? Okay? Now, if they are together, they can do even more bizarre things. Here, what you're looking at is actually rings of cells that are sticking to the substrate. So they stick to the substrate, and the substrate is formed in a ring or other geometries. They are dense, they are all together. I'm going to start the movie, and you will see very strange behaviors that all of a sudden they collectively decide to move one direction or another, clockwise or anti-clockwise, in a very coherent manner. It's as if all of you decide to go one particular direction. You know, you have a brain, you do it. Cells have no brain, yet they can compute together some collective phenomena of moving in a particular direction. Let's get further on this issue of movement. Who is swimming in the room, swimming in the swimming pool? Raise your hands. This is not something so, right. But you could see on the television people swimming in the Olympics, maybe, okay? So Michael Phelps, he swims at a speed of about 1 body length per second. He moves one of his body lengths per second. But look at other organisms like bacteria. They can move up to a few hundred body lengths per second. So rescaled to their body lengths, they move much faster. How is this possible? Okay? So, we're all too much familiar with our own behavior, and we tend to extrapolate the same to other organisms, but that is wrong. Cells and organisms could go much faster. Probably the faster multicellular organism swimming would be the sailfish, which goes at the 30 meters per second, which is 15 body lengths per second, like E. coli, but others move much faster. And here on the right, you see a movie of this bacterium, E. coli, swimming, okay? It moves around. It has this long kind of corkscrew behind. This is a flagellum to actually propel itself through the water environment. So, what is very strange about if you're a bacterium is the following. If you are on the pool and you dive in, what's going to happen? You plunge, and if you don't do any motion, you are going to move several body lengths, like maybe 10 meters, just with inertia. Your own inertia is enough to make you move through the water, because the viscosity of water is offering a very limited viscous force to resist your own motion set by your mass. So this is how you can move without any movement of your body. If you want to go further, you have to do some crawling and etc. But if you're a bacterium, your environment is like very thick honey. The environment for it is such that its inertia is basically zero, practically. If it stops moving, nothing happens. I put you here the number. It moves at a speed of 20 microns per second, which is 20 body lengths per second. If it stops doing its motion, it stops in 1 microsecond. If you do the math, meaning that it goes for a fraction of a nanometer, not even the size of a hydrogen atom, okay? So that means that there's no such thing as inertia playing a role when you're a molecule or when you're a cell. You should be surprised by that. Question here. No, you were stretching your arm, okay? This is a very, very different world to the world that we're familiar to. You know, we're familiar with a world where inertia, where mass, and all the associated effects are dominant in our behaviors. The living inside a cell made of many, many millions and billions of molecules is not like that. A molecule moving or a cell moving is offered resistance by the viscosity of the environment such that if it doesn't perform work, it stops instantly to move. So being alive is about having internal dynamics of your molecules. And the cells in an organism have to move. So they constantly have to burn energy to be in this motion because everything is super, super, super viscous, okay? This is a very strange world, a world that none of us can imagine, can experience, but you can only imagine. So for instance, in the bacterium, the way it moves is that it makes the rotation of a flagellum that you could see a drawing of here or you could see in the movie. It has this very complex machine where it's going to use chemical reactions to make the movement of this flagellum. Do you know the speed of rotation of this flagellum? 100 hertz. It is 100 rotation per second. Do you know one example of a structure that rotates at this speed? In an engine. When I came here, I took an airplane. At takeoff, you know, the speed of rotation of the engine is 100 hertz, round about that. So the bacterium, just to move, is doing the same over its many different flagella to just move through this very viscous environment. So this gives you a perception that the world of cells is very strange. It requires constant movement of the molecules, and that is made possible by the constant burning of chemical energy and its transformation into mechanical work, okay? So this was the puzzles of movement and dynamics in the living, okay? But now, even more amazing is the amazement at our own development. Each of you originated from a first allied egg that underwent its own development. So think of numbers. We humans are in a range of 1 to 2 meters, few tens of kilograms. We are composed of, you know, few 10 to the 13 cells. And at some point, we'll give rise to an egg, a single cell, 1 microgram in mass, 120 microns in length, in diameter, and then a new organism is going to reform. You should not, you know that, right? But you should not take it for granted. Because the question is, what is about this egg that is such that it is capable of regenerating a whole organism on its own? And you could say, well, I learned in high school or in whatever school you are that it's all about DNA. It's way more complicated than that. And even if you said so, what would you mean? So we don't understand really what is it about this very particular stage that we call a fertilized egg that is capable of transmitting all that is necessary to reform dynamically a new organism. If you think of, you know, image compression, you know, you could take a big file or a movie or a big photograph, you can compress it into a tiny file. When you do that, you don't lose. If you do it properly, you don't lose any information. You just remove any redundancy. You just keep the essential. So here there's something about like the algorithm of forming an organism and data that are compressed super, super highly, talking about the diversity of cell types, diversity of cells, all down to 1. These are the compression rates, if you want, in terms of length, size, and diversity. And all of that can be regenerated, okay? So there's something about development that you could regard as a compression algorithm, a very complex one capable of reforming each of you. And so that is one of the mysteries of life, one that I have been working for all of my career from the time that I was 22, and I moved to New York to start my studies, working on the development of the embryo of Drosophila, which was then and still is a very interesting and important organism to study development. Because I was truly amazed when I was your age by this very process of development. How could that happen? Okay? So this is, I think, one of the most challenging problems for biology, to understand the growth in complexity of our organism and the fact that it is so reproducible from one organism to another. If you look at this embryo on the right, it has probably in the range of a few billion cells. And if you look at 100 different ones, they would have very, very similar morphology. And any defect in morphology would have severe consequences for the health of the future organism. So this has to be done in a way that is accurate, although in the face of many, many perturbations or many fluctuations in the behavior of cells. Every single cell will not have an invariant process. I mean, think of you as a collective. You know, if you were to form a pattern outside, you would learn method number one, the choreography. Each of you would be assigned a particular task with respect to the other ones to form, you know, whatever shape on the lawn of the field that you may have outside. That would be maybe the logo of Ashoka University or whatever. That would be a very deterministic programming of your behavior. Do you think that in an organism, when one cell makes two and grow exponentially, having billions of cells, each of them is wired in a similar way to do a particular task? Who thinks so? Do you understand the question? You know, when I say you want to form a pattern together, it could be choreography. You could be programmed to do a very particular task. You will have no freedom, so to say, to do what you want. Okay? And if you failed, it would be seen by people outside. Do you think this is the way that development is happening? If you now transpose to single cells or many cells in a collective, in the back.
My question was that you mentioned in the previous section how one cell gave birth to so many different kind of organisms, right? So my question is, how did we all identify that specific cell? And does it vary for other people?
How do we identify?
How did we identify that particular cell? What was the method for the identification of that particular cell?
Okay, so to identify a cell...
And does it vary for other people, or is it the same for other organisms, or is it the same for every organism?
So to monitor cells in vivo, you could use methods such as microscopy. We'll go into that later on. You could really look at them. In my lab, we do a lot of microscopy, just looking at the process in vivo. When I started my career in biology in the early 90s, you could not do much of that. Or when you did microscopy, you could not discern a cell as it was living. You had to fix chemically the material. And so you could look at what molecule does it have in it. Okay, you could do an antibody labeling of a cell, but the organism was dead. What happened in the late 90s is that progressively it became possible through the development of microscopy and of fluorochromes, which allows to look at single molecules, to actually look at the dynamics of cells. And you'll see many examples in the next slides of those. And so this is a way you could actually track cells in vivo as they live and as they do the business of moving about and doing different things. That's the first method to track cells. And now you could do things like with the advanced microscopy as computational methods, like computational sciences allows you to track hundreds, if not thousands of cells in the environment of an embryo and to follow each of them doing their business. It's as if you took a picture of the city of Delhi, took an airplane, track, each car in the city and be able to actually learn from this the trajectories and the statistics of those, right? Okay. So this you can do, and this has changed tremendously. I was lucky enough to live through the revolution in microscopy, in fluorescence microscopy. You could do that for cells, and you can even do it for molecules. Some people could actually look at single molecules in a cell, okay? So that's the first method, and it has went through revolutions in the past decades, and there has been actually Nobel Prize for the discovery of GFP and similar molecules that allows to look at these molecules. This was a Nobel Prize in chemistry, and there was another Nobel Prize in microscopy to look at single molecules and things like that. And many other important things that will never get any Nobel Prize, but were equally important. The second method is looking into the chemistry of cells. Cells are made of RNAs and proteins and lipids and many sugar modifications and ions. The important things that you want to track would be RNAs and proteins, because these are a form of encoding of the chemical state of a cell. We humans have 3 billion nucleotides in our DNA. We have out of these few tens of thousands of genes. And in the human cells, you have, on the order of only a fraction of those that are expressed, that produce RNAs and proteins. And so you could actually get to the molecular identity of a cell by actually looking at its RNA content or its protein content. So it used to be that in the year early 2000, you could actually get the census of all the RNA species in a cell, but this was in populations of cells. Now what is possible is to do for each single cell, what are the RNA counts of each cell? So you could get its molecular identity. When you do that, the cells are dead. You can no longer track them, okay? So you get an information about the statistics of single cells in a population, okay? But this would be dead tissue. But you could now correlate also the information on microscopy and information of RNA content. So this is just a couple of methods whereby you could track cells and get to know their chemical identity and their physical status or dynamics. Does it answer somewhat your question?
Yes. Thank you so much, sir.
Does it explain things for you in the room, or do you want further explanation on that? So I have a question over here.
Sir, I have a question. So previously you showed us a slide of cells moving in perfect synchrony. You would put them on, I think, a circular plate and
Yes.
they were moving in perfect synchrony clockwise. So
Yes.
does this work with any kind of cell? Which kind of cells do you use for these kind of experiments? And also, how do cells bypass the Reynolds number?
How do cells?
Bypass the Reynolds number.
Oh, do they pass the neighbors? Huh.
The Reynolds number.
Okay, so in this particular experiment, I think if I remember correctly, these were cells from the kidney. But you could take a number of different cells, they would do the same. So long as they stick to the substrate and somewhat stick to their neighbors so that they made a coherent whole, okay? Cells can pass the neighbors occasionally, but in many instances, they don't. In particular, when you have chains of width, 1 or 2 cells, they just follow the neighbors. So they read their environment by sensing the neighbor in front and back. You want to say something else?
Sir, I'm very sorry. I think you misunderstood my question.
Sorry.
So basically, I was asking about the Reynolds number, the viscosity amount that is a quantity that is experienced by almost all cells.
Yes.
So I was asking how cells bypass the viscosity of.
Oh, how cells bypass their viscosity. Well, so the way they do it is because, as I said, they just consume energy a lot. They just, like the bacterium, for instance, is living in this low Reynolds number, which is a statement for the fact that they live in a world dominated by viscosity and not by inertia. If you want to move in a viscous environment with no inertia, the only thing you can do is just keep moving. Keep burning energy it's like crawling and crawling forever. So the bacterium has a flagellum, okay? And it constantly burns energy to do it at 100 hertz, right? So this is for the case of a cell swimming. If it is in the viscous environment of a substrate on which it crawls, it will use another kind of energy which is embedded in the cytoskeleton of cells. I cannot go into details. I don't have any data to show you, but this is well understood. It has some intrinsic, internal burning of energy to transform chemical energy into mechanical energy. And cells are very good at doing that. In fact, that's the main thing they do. They transform energy from chemical bonds, to mechanical work. Yes? Question here.
So I had a doubt regarding how the cells were wired to form life, because we saw that certain cells, it is more than chromosomes. So I know that certain cells can form body parts. At the same time, they can cause cancer. Other cells can help transmit chemical messages
Yes.
across the cellular surface when electrical signals are not able to reach every
Right.
part of the body. But the same type of cell can be cancerogenous in animals at a membranous stage or a.
So what is your question?
Yeah, so my question is, how does the same type of cell manage to do both these things at the same time?
Well, they cannot be cancerous and being in a good physiological state. They usually change states. So naturally, in the normal development of an organism, you have stem cells that then will differentiate into cells that have particular features, like a neuron, a muscle cells, etc. But sometimes, these decision-making processes that are very complex go wrong, and the cells, de-differentiate or acquires a growth potential and proliferation potential. That is what we call a cancer cell. Okay? This is just a byproduct of an otherwise extremely powerful, well-controlled process. But life is not perfect. And we have many means of correction, but sometimes cells go awry, and that cancer would be one of those. Okay, so rather think of it that cells can have multiple possible states. There are exquisite controls over the different state they can have, but sometimes cells manage to escape that, and that's what we call a cancer. Okay? Question, and then we'll have to move on.
Okay. So, sir, I wanted to ask that, for example, you observe these moving bacteria, these moving cells. So, I just wanted to ask that if you're separating them from some living source or something like that, how much time do they stay alive before literally dying off?
How much is a cell going to live from its environment before dying?
Yeah, like for example, a body cell. If we separate a
Right.
body cell from that organism, how much time is it going to live for before it dies off?
If you move it out of the organism.
Yeah.
So so you can actually take cells from some of our organs, put them in some culture, make what we call primary culture. If you provide energy source, chemical energy source, so-called growth factors, which are hormones, which we have in the body, that promotes their state and enhance their state of proliferation and growth, well, they could live for a long time, okay? I'm talking about if you're a good experimenter, you could keep them for weeks, probably, okay? It's all down to reproducing the chemical environment of the organism in forms of hormone and actually nutrition factors.
So basically you mean to say that if we can induce an environment similar to that of the organisms, giving it energy source and all, then we
Yes.
can keep it alive for as long as we like?
In principle, yes, okay? But there's something about, of course, you could take cells out, and if you understand well enough their environment, you could culture them and do science, okay? But the thing is that a cell in its environment is always going to be in a better state because it's evolved to be like this. There's something very mysterious about the organism that, as a collective, is way better than in isolation. And in fact, what is an organism? It's a collection of cells, like you're a collection of people. Each of you could have their own life, but as a collective, you would be much stronger. So that's an organism. In fact, it's the only way they actually truly survive and show their full potential. I use a lot of metaphors. As a society, we are individuals, cells are individuals, but collectively they do much better as individuals than if they were isolated. Yeah. That's a bit the idea. It's not that there is a tension or opposition between the two. As individuals, they do a lot. Collectively, they do better. Because why? They do an organism, right? And that requires a collective phenomenon. The living is a lot about collective dynamics of decision-making processes, about making a pattern, controlling movement. That's why I showed different examples of collective dynamics.
Sir, so that's why I believe it occurs through multicellular coordination. And it's not one singular cell controlling the embryonic development overall.
So maybe we'll go back to development later on, if you don't mind. I think your question will be maybe re-asked then.
It's not a question. You had asked a question in the start.
I can't remember what question I asked, sorry.
Sure, sir.
Okay, anyways. So I hope you get excited about development. Who is excited about development? Raise your hands. Okay. Who thinks it's boring? Be courageous. If there was no development, oh, it's okay. Okay. It's amazing because of the multiple, again, think about the choreography of billions of cells. How you do it. It is not by very exquisite wired programming of every single cell dynamics. They actually jiggle around in a way that is completely, subject to stochasticity and fluctuations. Nonetheless, in spite of all of them having individual behaviors that are very stochastic, they make something together that is highly, highly reproducible. That's the puzzle. Do you get it? This puzzle fact? Each of you having completely seemingly stochastic behaviors, but as a group, you do something very coherent. That's what happens in living organisms. And we do not understand that. It is still a very open puzzle. How do we know it is a puzzle? Well, exactly when we started, there was a question in the back about how do we look at cells? Well, we imaged them. And what did we observe? They had very stochastic dynamics. How do they form a very reproducible organism then? That's a problem. I don't have the answer to that, okay? So let's look at the beauty of development. This is here the development of a cell. It's a green alga that divides. Look at the very complex ornament of that cell. When the cell divides, it actually reproduces its own features, okay? These very complex structures at the edge is growing in front of your eyes and developing as the cell divides. So it can be very fast. Here at the bottom, you will see, what is it? A sea urchin embryo. It's a single cell. Something is happening. You see, it's fertilized and then it divides. 2 cells, 4 cells. That seems like a very stereotypical choreography. Every cell is going to do a very precise task. And in fact, if you look at these organisms, even later in development, it's highly reproducible down to the single cell. So this is not what, like I just said before. This is an example of very, very precisely controlled choreography of cell division in space and time. But later on, at some point, it will become much more variable. And if we look at the same thing for us humans, early on, after fertilization, it would not be that reproducible. It would be much more fluctuating, okay? So going to another scale where you will not see single cells, you will see actually, imagine here, it's a zebrafish embryo. There are tens of thousands of cells. What you see is a body structure that emerges in front of your eyes with microscopy. You will see here the brain, the head region, the segments of the body, and the tail. You would recognize the body structures, the features. This is animal development sped up. You don't see the individual behaviors, but collectively they will make a very, very nicely fit embryo that when it emerges from the egg will be able to swim and survive in the environment that it's born into. I'm showing you movies because I think it's the best way to look at development. You know what this is? This is the early steps of development of a chick embryo. We eat eggs, those who are not vegetarian, and you see that this would, if you were to use a microscope to look on the surface of the egg, if it's fertilized, this is the anterior head region, tail region of the animal, and you see these flows of cells that are very organized. Can you see the flows of cells on the movie? You cannot discern individual cells because there are too many of those. Okay, although with this microscope, you could actually go down to the single cell. You could zoom in like a Hubble telescope and see it. It's not on that movie. I removed it. Okay? But you could see here and track. There was a question in the back about tracking many cells. You could track the cells. Collectively, statistically, they make very invariant features of vortices, okay, like a very organized fluid. But down to the single cells, you would be subject to some fluctuations. It's a good illustration of this cohabitation of fluctuations and yet collective dynamics. This is my beloved fly embryo, Drosophila embryos. These are the nuclei you're looking at. Again, down to the single cells, you can find statistical regularities, and you have collective dynamics that is super regular, but single cell will not have exactly the same behavior, okay? Who thinks a fly embryo is a superior organism because it's so beautiful? I raise my hand. Okay. This is very subjective. All organisms are great. And now there are more organisms than there used to be. You know, so-called model organisms are organisms that have been chosen for one reason or another by people over the past century to be worthy of investigation because they are easy to culture, etc. Drosophila, for instance, it was Morgan, T.H. Morgan, who switched to Drosophila in early 1900s because it was easy to raise, it was cheap, and he could do the genetics. In fact, he discovered chromosomes. Chromosomes were known, but he could actually get to the study of mutations and relate them to chromosome structure in the early 1900s. And since then, it has been used at many levels for the understanding of genetics, and then development, etc. So it's a great organism. So now we are looking at single cells. Could you actually see here the features inside cells? We'll come back to that. It's alive, it's dynamics. You can see fiber structures in green, and you can see other molecules in purple or orange. I'll come back to that later on. This is the sort of movies that we took in my lab. It's every second. We could even get a Z-stack, or like a volume, every second in the embryo. So we could really compute two colors of two molecules in a volume every second. So we see like almost like the real-time dynamics of molecules and cells as development happens. Okay, I want to show you something that is even more puzzling, and we'll close the chapter on the puzzling. Error rate. So do you know that, who knows what DNA replication is? Raise your hands if you know what DNA replication is. Otherwise, I will explain. So some people don't know. So in our genome, we have DNA, and we need to duplicate DNA every time we divide, so that we make two copies that is split into two daughter cells. Do we have to do it accurately or not? Because if we don't, that means we're going to send the wrong message to the daughter cell. And since there are going to be a few tens of cell divisions in an organism, if we fail, there will be many disease states like cancer. So we cannot not have exquisite control over DNA replication. So what is the error rate of DNA replication? It's shown here. 10 to the minus 8, 10 to the minus 10. That means that the enzyme, the molecule that is going to duplicate DNA, is making an error every, on average, every billion nucleotides. We have a few billion nucleotides in our genome, 3 exactly. That means on average, once per cell division. The fact is that most of our nucleotides could be mutated and they have no effect. So that's why there are no defects with that. It's a very low number. So I give you a sense of how low this number is. Think of the DNA replication that is a complex enzyme that is moving along DNA. You know that DNA is made of nucleotides, A, T, G, C. Did you know that? I think so. So think of this as a metaphor. During replication, the macroscopic replisome, this enzyme, think of a truck, travels at a speed of 500 kilometers. This is all in proportion, okay? It doesn't move really at 500 kilometers, but if it were to move at 500 kilometers an hour, it would make a delivery of 1 or 4 colored boxes on both sides of the street every 10 centimeters. So think of the truck delivering, every 10 centimeters at the speed of 500 kilometers an hour. It completes its journey in bacteria in 40 minutes. And having an error rate of 10 to the minus 8 to 10 to the minus 10 is meaning that in this highly efficient delivery process, the truck would deliver a wrong package only once every 3 years. 500 kilometers, both sides of the streets, every few centimeters, it makes an error in delivery only every 3 years. This gives you a sense of the very low error rates that has made our life possible. And this is a problem that was sorted out long time ago, and that allowed the evolution of life. Don't you find this amazing, these numbers? This is just a way to get you, the way you could be most amazed in life is because it's beautiful, but also when you get a number that surprises your intuition. And much of what I've said and how we started is about that. Get your intuition, work through your intuition, and then get the numbers by either making a calculation or making an experiment, and then realize, oh, that's really off my intuition. That is the beginning of a discovery, okay? And I think it's true in general in your life. We live in a complex world. We're talking a lot about ecosystems and the evolution of ecosystems because of climate change. Get the numbers, think critically based on numbers, but work through your intuition as well. That is a lot the method that I'm trying to suggest to you. I hope this goes through, because beyond the examples, this is what I want to convey. And that's where we started with this quiz question.
My question was the first slide of morphogenesis showed how one of these cells was reproducing and it was regenerating its lost parts, right? That got me thinking if this could be applied in biology, in medicine, for regenerative medicine or maybe like organ regeneration. So, is there any scope for that?
Is there any what? Sorry, I did. Scope for what?
Scope for regeneration or regenerative medicine?
Okay, so is there a scope for regeneration? Yeah, so I can only provide a quick answer. Well, regeneration is the natural property of cells in an organism that, you get wounded, you cut, it regenerates. You cannot regenerate a whole finger, so not everything regenerates. You could regenerate your liver, you cannot regenerate your brain. So cell organisms have evolved a strategy to regenerate. You could also use stem cells to regrow, providing the right substances to actually get to make an organ in vitro. This is a strategy based on stem cell research, which has exploded in the past 10 years and is a subject of intense research these days, that you could really do in vitro some kind of reconstitution. And so that allows also to study in culture things that otherwise happen deep in the wound, in the womb, where you would study an organism otherwise, and you can't, especially in humans. Okay, that's all I can say about that at the moment. So about the amazing, the last bit I want to say is something I already alluded to, is the energy. Okay, we are made of 10 to the 13 cells, 10 to the 13 cells. We burn every day on average 2,000 kilocalories. Okay, the task that we have to do is to deliver this energy to every cell. Some cells can survive without oxygen for a little while. They have developed a strategy to, be in this state. But most cells, especially our brain cells, if they don't have oxygen, they will die quickly. And in fact, if you have a stroke or if you have, if you drown in water, the first damage will be seen is in your brain. So the demand on energy of a cell is huge. I'm giving the number on the right. On average, think of 30 million molecules of ATP. ATP is the currency of energy, chemical energy in a cell. Okay? This is it. ATP is a small molecule. Who knows ATP? Okay, you all know ATP, right? This is a chemical energy, okay? It's the currency that you want to have. So, do you have a sense? So 30 million ATP molecules are burned in a cell per second. Okay? Do you have a sense of, in 1 hour of lecture, how much ATP in mass you burn? I'm asking the question again. In 1 hour, okay, how much ATP you burn? Sorry? You could make the calculation because I gave you everything here, but you could get a sense of the mass. Who votes for 1 gram? Who votes for 1 kilogram? Who votes for 1 ton? Okay, who votes for less than a gram? I don't see many raised hands. Okay. Well, the thing is the following. You will be surprised, hopefully. In 1 day, you burn about half of your body weight in ATP. In 1 day, you burn about half of your body weight in ATP. Who is stunned by this number? Raise your hands. Good. I managed to stun you. Okay? This is a surprise of numbers. These are the basic numbers, but you could recalculate from that what I just told you. Okay? It's a huge number. So to stay alive requires a constant burning of chemical energy. I gave you examples. If you want to move, this is another task. If you want to divide, this is another task. If you want to just stay alive, to move your internal body parts in the cell, requires constant energy flux. It's a huge number. For us, it's 2,000 kilocalories a day. You could convert into watts. Okay? It's 50 watts. Is 50 watts a lot or not a lot? If you ask like an AI machine to do the kind of very complex tasks that we do constantly, okay, even half conscious, okay, how many watts would that be? Thousands of watts. So we are way, way, way more efficient than anything else. So we are amazing machines at burning energy in a super efficient way to do very complex tasks. But nonetheless, that means that we burn half of our body weight of ATP every day. We recycle it. It goes from ATP to ADP and back, okay? So this is one of the mystery of life, that it requires like a huge energy flux, energy flow. And this is something that, you know, researchers are working hard on. So life is beautiful and amazing. I want to now go into something else. Life is diverse, but we'll show that behind this diversity, there is unity. This is a very important aspect of research investigation, and I want to get into that. Are you ready? So life is diverse. Look at these many organisms. Their shape is so different. Cells are very different morphology, all crazy structures. Nonetheless, if you compare, you find these amazing regularities, like polygonal shapes pop up every now and then on the shell of turtles, on coral reefs, on fungi, even on cells of our body. Spiral forms emerge at every scale, from plants to shells. Stripy patterns likewise. So how is it that these recurrent so-called universal geometries emerge in the living in materials that are very different? Between the spiral of a shell and the spiral of a pine cone is very different. Nonetheless, it makes these very regular mathematical structures. So this is something that is quite puzzling. The flocking behavior or the herding of sheep is quite remarkable. You've seen these drone movies of different animals, and there are plenty on YouTube. I'm not spending much time, it's beautiful. But the flocking of these animals could be likened to the flocking of bacteria. And the flocking of bacteria, shown here on this movie, could be likened to the flocking of single molecules. So what you see on the left is a cartoon of these filaments, are so-called actin filaments in the cell. They are a dynamic scaffold of the cell. Okay? They constantly turn over and move, and they are moved about by so-called motors, which are shown in orange. The small motors in orange, they power the scaffold to make it contractile. If you look at any cell, you would find that it has pulses of deformation. Can you see these pulses? Like accumulation of this color in some places and local tissue cell deformation. This is because they burn ATP and there are motor molecules that deform the material. So in every single cell in your organism, you have this constant pulsing of life through the dynamics of cells. And this is an example of the conversion of chemical energy, ATP, into the mechanical work that deforms the material. And that is done by this beautiful motor. This is my favorite molecule. It's called myosin. It's the molecule that we've worked on for so many years in my work group in Marseille, because this is thereby that you can explain how cells can contract. So this I would call molecular flocking or swarming. Okay, it turned out that between molecules, cells, and organisms, there are similar collective dynamics. Yes?
Yeah, in this video where they are actually overlapping and we could see a yellow signal, this is the overlapping with actin with myosin at that particular point. I mean, what are the proteins that are staying?
The signal in blue and
I'm
orange,
just exactly saying there, yeah.
This is myosin. This is a myosin molecule.
So myosin, when getting overlapped with actin, we are getting yellow signal.
It is actually walking on actin filaments.
I see.
It's walking on it, and it moves its heads, and as it does so, it actually makes the contraction. If you could use a molecular drug, you inject in it, within a fraction of a second, the motor is dead. It doesn't burn ATP, and you know what happens? Everything is frozen, dead. The molecules are there, but it's like you unplug the computer. Nothing happens.
Understood.
By just blocking the ATP hydrolysis on the motor, or something that is a proxy for it, which activates the motor, its capacity to do it.
And I was just wondering, when this live cell imaging has been done, I mean, I could not see nucleus all around. I mean, these are the cells.
No, because this is just a surface view of the cell. The surface that is pulsing and contractile, but not the inside, not the nucleus and all of that.
I see.
That's why. It's just a little surface view.
And
this requires very advanced microscopy. This was not possible even 20 years ago. This kind of technology, to see it, has advanced tremendously.
This has been performed on a high-end confocal microscope or
Yes.
some other...
Confocal microscopy.
Understood.
So this is just to say that, you know, the similarities amongst many things in the living, you know, is seen here. Similarity, this universality of shape, the universality of collective dynamics, of swarming or flocking if you want. This is based on the following principles shown here. Active motion, these are self-propelled entities, whether a sheep in a herd, or whether a cell in a tissue, or whether a myosin molecule in a cell. These are entities that are self-propelled, okay? They use energy to be self-propelled. They have properties of interactions that could be attraction or repulsion. And third, and not least, they have the so-called ordering property, alignment. It's not enough to be a sheep to move. You want to do like your neighbors. You want to correlate your dynamics with that of your neighbors. And that is true of cells in their environment. You remember the cells crawling together in a very coherent way because they correlate the dynamics to the neighbor. In fact, when you go into a very crowded environment, you know what is the best strategy to go through a crowd? Is not to try to be smarter and to go faster than the other one, because that creates jamming. What you should do is just follow your neighbor very close. If you do it, it will fluidize the tissue much more. That is an insight that comes from studying the physics of those collective phenomena, okay? Think of it next time you rush for taking the bus or whatever, okay? So what is the source of unity in the living world? Well, could be one reason why so many things are so similar despite very different morphology, okay? However, I want to get into something which you may not be that familiar with, but is what I would like to finish talking about today, which is the generality of physical principles. The physics is the same. You know, generally, when you learn biology, you learn the making of a cell, the making of an organism, the chemical composition, the central dogma of DNA makes RNA, which makes protein, and all of that. That's true, but we don't talk about the physical principles of cells. Cells are physical objects. They have properties that are compatible with the laws of physics. There's a particular physics in the living that is peculiar in its way of functioning. It's not that you require new forces, but the collective properties of the living call for a new physics, if you want. In particular, this property of being an active material or an active fluid is different from the standard fluid that we know of. If you take honey or if you take water, you know, this is a passive material. You could subject it to an external force, but a living material has its own energy consuming process that allows its own activity from within. Okay? I don't have much time to elaborate on that, but it's so-called an active fluid. That was something that physicists coined and studied in the past 20 years or so. Okay? So I want to go now to the generality of the physics of life, and I will illustrate it on two basic phenomena. Are you ready to go into the physics of the living? Raise your hands if you want. Else you go. No, I'm just kidding. We stay on board, okay? Is it the time you want to ask your question, or? Okay, quick question, quick answer.
What happens when there's an error in DNA replication?
When there's an error in DNA replication? Well, if you hit randomly in the genome, you will not be in a gene. So it will have no effect. If you hit a gene, you want to hit it in a region of the gene that matters. Again, not every nucleotide matters. So the fact is that most mutations have no effect. It's not like, by the way, a computer. Who's programming in the room? Who's writing code? Raise your hands. Who is a computer geek? Raise your hands if you code. You know so-called bugs. If you have a single error, like if you do a random mutation in your language, the whole thing will stop, most likely, right? It's very brittle thing. In the living, the algorithm of the living is very robust to such perturbations, okay? This was recognized a long time ago by people like John von Neumann who developed the first principles of computers, comparing to what might happen in auto-replicating automata called cells and organisms. It's different. And so your question is hitting that. Not every mutation has an effect. Good for us. There's a low error rate. This will not necessarily translate into a mutation in protein sequence because of the redundancy of the genetic code. And thirdly, most mutations have no impact because, you know, close amino acids mutated will have similar chemical properties. same charges, same hydrophobicity, for instance. Fourth, they will be in regions of genes that don't even matter, or even outside of genes. You know, most of the genome is not genes, okay? So it doesn't matter. So that's why, you know, most of the mutations have little impact. There's few mutations and they have little impact. Sometimes they do. We can even correct for those. The cell is going to be killed or is going to be phagocytosed. If it's a tumor cell, that happens. But every now and then we have errors, and that's why, you know, eventually we can get a disease, but we have doctors, okay?
Good afternoon, sir. I just had a question regarding the cells. So my question was like, how do the cells like of an organism like interact with each other and like with like the same organism and like another organism? That was my question, sir.
Okay, you're asking the question of the language of cells. What language do they speak? In India, you speak many languages. Who speaks more than 2 languages? Raise your hands. Who speaks more than 3 languages? Wow, still a lot. 4 languages? 5? Wow, that's impressive. Anyways, so you speak many languages. You know, if you did that in Europe, there would be at most 2 languages, maybe 3 occasionally. So cells, how many languages do they speak? Well, it depends how you view it. They can all speak to one another. You know, they exchange chemicals. But is the language of the living just chemicals? Who thinks the language of the living is mostly chemicals? Raise your hands. It is mostly chemical. This is what we learn. But what is another form of language in the living? Electricity, yeah, that's a language, I agree. Another language? Well, that would be chemical world. Other language? I want to hear one word. Mechanics.
Mechanics, yeah.
Mechanics is a very important language. But, you know, physical forces generally, you know. All life is about physical, chemical processes. Okay, so let's move on. So I want to say a few famous quotes. These, for me, capture the essence of why studying the physics of life, okay? This guy was a very famous mathematician, Henri Poincaré. Okay, you may know his name. I translated what he said in La science et l'hypothèse, Science and Hypothesis, written a long time ago. The scientist must order. Science is made of facts, as a house is made of stones, but an accumulation of facts is no more a science than a pile of stones is a house. So the work of a scientist, like I have been, has been to try to look for order. It's a beautiful quote, don't you think? The ordering mission of scientists, finding universality behind the diversity. This has been the mission of scientists from the time they started to give names to animals, understanding what's beneath that. An even more remarkable quote from Jean Perrin, who got the Nobel Prize in Physics for his work on Brownian motion. Okay, he proved Einstein's foundation of Brownian motion, doing experiments. And so he wrote a book called The Atoms in 1913. And in that he said, the science replaces the complicated visible with the simple invisible. It's almost like a poem to me, you know. What is the simple invisible? Well, for him, these are the atoms, obviously. But metaphorically, I would say the simple invisible is a world of models and theories and conceptual models that we use to capture the essence of the universality behind the living. Okay? To me, this is like one of my, you know, important quotes that, you know, guide me in doing science. Of course, we peer into the diversity of what we observe, but from that, we try to see what are the regularities, what is the universality beneath that. And to get to that, you need to understand the physics of life. So this regularity has been long proposed to be understood using the language of nature, which is, according to Galileo and many people since then, mathematics. Let's read him. Philosophy, which for him is physics. You know, philosophy is basically a study of natural philosophy and so physics. Is written in this grand book, I mean the universe, which stands continually open to our gaze, but it cannot be understood unless one first learns to comprehend the language and interpret the characters in which it is written. It is written in the language of mathematics. So this is the language that they speak, you could say, not just molecules and mechanics, but fundamentally mathematics. And its characters are triangles, circles, and other geometrical figures without which it is humanly impossible to understand a single word of it. Without these, one is wandering around in a dark labyrinth. So I'm saying that if you want to study the living, you have to know mathematics. not, like a professional mathematicians, just, mathematics just to understand algebra and other things. Physics, chemistry, computational sciences is very important. These are all important languages. And the last quote I want to have with you is from Schrödinger. He wrote this very important book called What is Life, in which, as a physicist, one of the fathers of quantum mechanics, he thought about the amazing world of the living. And he said, how can the events in space and time which take place within the spatial boundary of a living organism be accounted for by physics and chemistry? And he said, we cannot do it now, but the obvious inability of present-day physics and chemistry to account for such events is no reason at all for doubting that they can be accounted for by those sciences. So I'm saying to you that yes, physics has provided deep answer to the mystery of the living, the mystery of its dynamics, and the many puzzles and amazing things that I showed you at the beginning can find deep answers by studying the physical foundations of the living, okay? And that adds to what you've learned at school about, the molecular chemistry of the living, which is what we learn, but this is like learning a dictionary without knowing the grammar. You learn a language, you have a dictionary, you have a book, you learn the words. We need to learn the grammar. The grammar is all these invisible, simple things that is the conceptual models. That you learn step by step. But the way we usually learn biology is mostly based on the catalogs of genes and cells and things, which is hiding the importance of grammar and the syntax and all of that, okay? So, I'm going to elaborate on two principles of physics that turns out to be very important if you want to understand development, animal development, okay? And even, I would argue, for plant development. The first one is bubbles and foams is non-living objects, and how principles that explain bubbles and foams could be used to understand cells and tissues. Let's go into that. So let's look at these cells. This is another of these cells called ciliates that swim in waters. They have the cilia to eat other ciliates, like you see here. This is a so-called stentor cell. It has this very particular cone shape, which is like a big mouth open to eat. This is a very complex organism that has memory and learning and predation and all of that. This is a drawing which is from Darcy Thompson in his very famous book called On Growth and Form, 1917. And in the 19th century, there were many people, in particular this guy called Kent, who observed with simple microscopes the diversity of these cells, which all look like flasks. They look like objects that have very regular geometries. And this was puzzling to them. These are called protists or infusories back then. And so there is a physicist called Plato who's thinking through these particular regular shapes, okay, of cells, thought of maybe a way to understand it from the physics, the statics of liquids. He argued that, the shape of a bubble, okay, is dictated by its energy. If it wants to minimize its energy, it would minimize, say, its surface. And it turned out that the mathematics of minimal surfaces is well studied, and there are not an infinite number, actually it's a very finite number of finite minimal surfaces. The sphere is one, obviously. There are other ones, like these hyperboloids or helicoids as well. So these minimal surfaces are found in the living because they usually correspond to minimal energy of the objects. Okay, so the regularity of the geometry is dependent upon the tensions. If it is the same all around, the minimal surface would have this particular conformation. You could transpose that to the structure of, the honeycomb patterns in beehives. They warm up the fluid, it will cool, adopt the minimal energy, which is a minimal surface. It makes exactly 120 degrees. It's not that they have a compass to measure it. Physics does it for free. Bees do not have a brain to measure this particular mathematics. It just comes for free because it's the physics of the living. Minimal surface, minimal energy. So for instance, this is a blow-up of these particular images from the book of Darcy Thompson. Some veins and others have different angles, presumably because the tension that guides them is following these particular rules of equality or inequality as shown at the top. This was the sort of language that Darcy Thompson used. You know, he was a fervent believer that mathematics govern the world, like Galileo. Yes, question?
Yeah, sorry, about this itself. So you talked about the example of bees and the honeycomb shape, right? So you're telling me that the shape of the honeycomb comes spontaneously and not through the knowledge of the bees?
If you knew the knowledge of the, if you knew, so your question is, sorry, maybe I missed your question. Could you?
So you're telling me that the shape of the honeycomb, it comes due to its, due to the shape adopting a minimal surface area for minimal energy consumption or?
What the bees do is that they warm up the wax by using their wings beating. So the temperature goes up and the wax melts, it becomes a fluid. And as it will cool, it will form these more regular structures. And they also have inside, you know, the little, you know, the nymph, there's an object inside which is the developing organism, which makes a constraint on the structure. Okay, I'm not a specialist of beehive, but this is a general feature that the physics of a cooling fluid that goes through a minimal surface on the constraint of having inside an object of regular size makes this so regular. It's not that they compute and measure anything. That's the central message that I wanted to say. Okay? So, but now look at embryos, because the same principles of foams and fluids can be used to study much more remarkable things than shapes of cells. This is a frog embryo undergoing the thing that started your life, which is gastrulation. Gastrulation is this process whereby collections of cells in an embryo form a complex 3D object. Here you see the neural tube forming. Down in the groove you have the future brain cells, or the actual nervous system, here toward the head region. Okay? The spina bifida is a disease of that particular process. It doesn't close normally, and that's, as you know, has consequences. So that is what you can see in a movie of gastrulating embryo. This guy, Johannes Holtfretter, who was in Germany and moved to the US during the war, he did experiments that are very famous. He took this so-called neural fold structure of a gastrulating amphibian. He took the neural plate and he took the epidermis. These cells had different colors because they had different pigmentation. He used chemicals to dissociate the cells, so they would form dissociated epidermis cells, dissociated neural plate cells. He let them reaggregate and waited. And what he could see is that this forms a reaggregation with salt and pepper structure, but following about a day, I think, these would form structures where the cells of different developmental origin segregated from one another. They're so-called sorted out. If you make a section through it, that's what happens. The cells were, you know, it was salt and pepper at the beginning, and gradually the cells of the epidermis of the neural system would form a core surrounded by a shell of epidermal cells. How do cells know how to do that? What language do they communicate with one another to do it? Well, this could be well explained by the language of surface tension, which is something which is well used to characterize fluids sorting from one another, like oil and vinegar. So let's go into surface tension. This is a cartoon representation of a fluid where you have here oil and here water. The oil molecules interact with one another and they love it. So they have, like, on average, you know, uniform force of interactions amongst them. Of course, this is subject to fluctuation, so they move about, but on average they will not. Water do the same, but at the boundary, the water molecule, the lipid molecules will interact non-uniformly with the neighbors. As a result of that, there will be imbalance of the interacting forces, and so they will be moving away from the boundary. If all the molecules in the boundary move away from the boundary, they will create a tension. That is a surface tension. It's an emergent property of non-uniform interactions amongst molecules at the boundary, because the molecules, you know, a hydrophobic molecule will not like to interact with water molecule. This is the essence of surface tension. You could explain how a drop of water will make on the surface a particular geometry, and the angle reflects the importance of surface tension. If it has low surface tension, water will spread on the surface. It is called a wetting force. It wets the material. If it is subject mostly by surface tension, it will round up and say, I don't want to spread around. The alcohol will spread, okay? Water on some surface will not. On these leaves, water is meant to not spread. So this is exactly what happens in these materials. If you take the cells, you could make experiments to measure the surface tension. The surface tension is what relates the energy change that is associated with the surface change at the interface between two fluids, okay? And so here, the cells have the same properties, they have the same chemical stickiness to the neighbors, so they mix up. Here, they have a difference. The red cells like to stick to one another more than with the green cells, so they sort out. And it is possible to measure the surface tension, and these are these numbers here. And predictably, difference in surface tensions make enveloping behaviors just like fluids do, like molecules, like water and vinegar, like oil and vinegar. So this shows you that you could use the language of mechanics and surface tension to explain sorting behavior. Okay? Are you with me? Questions on that before we finish on the last chapter, which is short, which is active fluids. It's not just a passive material like oil and vinegars and this fluid. It's an active material. We talked about that much. These are embryos that gastrulate. So they change their curvature. How do they change their curvature? By using active forces. So we introduced the material before. This gel at the surface of cells. I showed you movies of these filaments that move about because they have motors. And they produce mechanical force within cells. And later on, I'll show you how you can measure these forces. Okay? Because we can measure these forces. And forces means that they're going to deform the cells. You see here, in green, is actually this motor molecule that deforms the cell. It accumulates into clumps that deforms the cells. Here's another analogy with fluids. Cells move about. This is one of the things that we did in my group in Marseille about 20 years ago. We looked at an embryo that extends its axis, and we found that this fluid is not a passive fluid. It does it in a very coherent way of reordering contacts, and this is the transition in the topology of the interfaces. And this is done by a motor, but soap bubbles do the same if you provide an external force. Look at these soap bubbles. Some contacts disappear, and new forms. It's a change in the conformation. And this is done by active forces shown in green, like these cables here. These are active tension that reorganize all the contacts between the cells. Okay, and I will finish with just a couple of ideas of how could you measure mechanical forces. Do you know how you could measure forces in cells? If you want to test, is there tension in these cells, like I'm saying? There's more tension here. Do you know how you could measure forces? Do you want to know? Okay. So the first thing is that imagine you have a spring and you have a tension. If you cut it, the speed at which the spring will relax will be indicative of the forces, the tension it has, okay? So this is the same we did. So I teamed up with a physicist who actually came to me to say, he saw our paper where we proposed there was an isotropic tension. The tension was organized spatially, but we didn't have any number. It was still a hypothesis. We know it used a motor that does tension, so how could we measure it? Well, in our collaboration, we developed a laser that could do so-called nano dissection. It could break within cells this actin mesh that I showed you, which is shown here, actin myosin. We break very locally this mesh and look at the relaxation kinetics of the cells. You're going to see in the movie the relaxation kinetics. You see the flash of light, and then you see it relaxes. Well, this is something else. We showed there was no hole in the cells. We didn't kill it. It was perfectly happy. But you see the flash of light, and then this relaxes. We could measure the relaxation kinetics is in particular the instantaneous relaxation at the beginning. This is proportional to the tension divided by the friction in the environment, okay? And so from that, we could actually measure how much tension there was there compared to the other ones. And it was about twofold. Then another method could be used. We used, so that's actually in my collaborator Pierre-François Len. They used a so-called optical tweezer. You could use the properties of light to pinch an object when the refractive index is different from the environment. And so you could trap the edge of a cell here. And can you see the oscillation? Because you move the beam, and so you move the properties of the cells. And so it's like, you know, you have a guitar, and you take the string and you pull it. How much tension you have is going to form a particular deformation. So by measuring the deformation of the edge and knowing the properties of the trap, the tension, the coupling strength it has, elastic coupling strength, you could measure the tension. And there we had even the number of newtons, okay? So with that, you could actually measure very precisely the forces inside an organisms, okay? So that's the end of it. I wanted to show you that the unity of the living is certainly explained if you understand the universality of genomes and due to Darwinian evolution, but the universality of this invisible language of physics, of the principles of physics written in the language of mathematics is one of the reasons why you could explain the universality of properties in the living beyond the diversity of the observation. And I will leave you with, someone was asking about information. The flow of information among cells, you could regard it in two ways. The language is going to be made of genetics and biochemistry, mechanics, and something else I don't have the time to get into, which is geometry, the object properties where this happens. These are the languages the cells use, okay? But you could see it as a program. The information flows from some storage or initial condition and cascades down, much like you would build a building. You have a plan, you have an architect, you have modules of manufacturing, and you follow a chart. That's one way of thinking about it. But another way is inspired by the termite mound that you find in India, for instance, which is called self-organization. In that, we have the same modules of information, but we say there's no such a thing as an architect, there's no hierarchy, there's no so-called deterministic plan. It is a world of stochastic fluctuations, which is exactly what we know of molecules and cells. But the properties of the interaction are such that patterns emerge naturally. When an ant colony forms, there's a queen, that's for making the eggs, but there's no architects of the ant colony. Same for termite mounds. It just emerges naturally. The self-organization is what could be used to explain chemical world like these instabilities in vitro. Maybe you heard of that, or Turing instabilities. But this is happening everywhere in the living, and I will finish with one particular. So this can be used to explain the patterns of shells, for instance, the same fundamental properties of propagating chemical activities in a tissue. But look at these neurons, and this will be my last slide. This is a neuron that people in my lab study. It has very complex morphology, and its complex morphology is arising from stochastic fluctuations in the branching and growth dynamics that you could see here. You could use computational methods to actually track the dynamics. If it's growing, it's going to be green. If it shrinks, red, and otherwise it's going to be blue. So from that, you could learn the statistics of these self-organized systems. I will leave you with that. Think of the world as a very dynamic world where the language, the algorithm that underline the formation should be seen as an interplay of these two modalities of information flow. Program, which is top-down, or bottom-up, which is self-organization. And the two coexist within cells, within tissues, across scales, to explain these many phenomena that we looked into at the beginning and which I refer to in this section on the amazing and puzzling aspects of life. So that's it. I hope I conveyed to you a picture of the breadth of biology and why we need people from every horizon of science, from biology, chemistry, physics, computer science, mathematics. Thank you very much. I'm happy to take more questions, but I've been already overboard.